Hello, welcome to Bite Size Med. In this video, I'm going to be going over some of the basic stuff that's needed to understand the action potential. Action potentials are how excitable cells, like nerve and muscle cells, transmit signals, which is basically information. Nerves use action potentials to transmit signals from the periphery to the central nervous system, or the other way around. In between the action potentials, the neuron is said to be at rest. So the membrane potential during this state of rest is called the resting membrane potential. This is the potential difference across the membrane, the inside compared to the outside. So the outside is used as reference, and the numbers we talk about are on the inside of the cell near the membrane. For large nerves, that resting membrane potential is around negative 70 millivolts. So the inside is more negative compared to the outside. Let's quickly go over how this negative resting membrane potential is maintained. Inside the cell, potassium is higher, and outside, sodium is higher. The membrane has leak channels. Leak channels allow ions to leak through. They are diffusion channels, so it's along the gradient. Potassium leaves the cell and sodium enters. But there are more open potassium channels than sodium. So at rest, the membrane is more permeable to potassium than sodium. That's why the RMP is closer to the equilibrium potential of potassium, which is around negative 90 millivolts, compared to sodium, which is positive 60. Negative 70 is closer to negative 90. I'm going to be using round numbers like this. Different books give different numbers. I just chose these to make it a little easier to understand. Inside the cell, there are also negative anions, like proteins and phosphates. They can't leave the cell, they are stuck inside. More positive ions are leaving the cell than are entering, and there are negative ions inside. So you can see how it's making the inside more negative than the outside. There's also a sodium-potassium ATPase pump. This is an active transport pump. It moves ions against the gradient. When one molecule of ATP gets hydrolyzed, 3 sodium ions are sent out and 2 potassium ions are brought into the cell. Again, there are more positive ions leaving than entering, and also this pump helps maintain the concentration gradient. So the resting membrane potential of a neuron is negative. Now this potential is going to briefly change during the action potential. How can the potential change? Again with ions, sodium and potassium. Now there are voltage-gated sodium channels and voltage-gated potassium channels that come in. These are different from those leak channels that we saw at rest. These channels are voltage-gated, meaning a change in the membrane potential is what opens and closes them. Let's take the voltage-gated sodium channel first. This channel has 2 gates. The one on the outside is called the activation gate, and the one inside is the inactivation gate. These gates open and close during the action potential. At rest, the activation gate is closed and the inactivation gate is open. So ions can't pass through. These are closed channels. When the activation gate opens, the channel is in an open state. Now ions can pass through. At the peak of the action potential, the inactivation gates, those start closing. Again, ions can't pass through. These channels are inactivated, and after a short duration, they will come back to the closed state again. This happens during the action potential. The closed channels open and then get inactivated. So the closed channels can be recruited to open up and let ions pass through, but the inactivated ones cannot. They have to come back to the closed state first. The voltage-gated potassium channels are a little simpler. They just have one gate on the inside. When it opens, ions can pass through. When it closes, they don't. So when the channels open, ions can pass through. Which way will they move? What drives the ions to move is an electrochemical driving force. They want to bring the membrane potential close to their equilibrium potential. So when the sodium ions can move, they try to bring that negative 70 towards positive 60. How can it do that? How can it make the membrane potential more positive? By entering the cell. This is a positive ion. So that's what sodium does. It enters the cell. What about potassium? Negative 90. they have to bring that negative 70 towards negative 90. To make it more negative, how would these positive ions move? They will leave the cell. So when the channels open, sodium ions enter the cell and potassium ions leave the cell. If the membrane potential were to cross the equilibrium potential, the direction of movement of that ion would change. That's why the equilibrium potential is also called the reversal potential. Now, we saw at rest the inside is more negative than the outside, and remember this is happening just at the membrane. The overall compartments maintain that bulk electroneutrality. So the membrane at rest is polarized. Let's take a small portion of the membrane. The orange is inside the cell and the blue is outside. And at rest, the membrane is polarized. When sodium starts entering the cell, it's making the inside more positive. That is depolarization, when the potential becomes less negative and moves towards the positive side, like from negative 70 to negative 50, or it can even become a positive number like positive 30. That's depolarization. When sodium stops entering and potassium starts leaving the cell, then the potential becomes more negative. It's coming back to the original negativity, that is repolarization, like from positive 30 back to negative 70. So it's becoming more negative. What if it becomes more negative than that, like down to negative 80? That's hyperpolarization. So depolarization, repolarization, and hyperpolarization. Those are the important phases of the action potential, and in part 2, we're going to see how that happens. If this video helped you, give it a thumbs up and subscribe to my channel. Thanks for watching, and I'll see you in the next one.